All right, good morning, everybody. I hope everyone enjoyed day one and the workshops when you were here. Today, day two of talks. We are going to start off with Paul Thomas. He's from the University of Southern California. He's the PI on the gene ontology project in Panther, and I've known Paul for a very long time. I've been learning about the latest and greatest in protein function prediction for over a decade from Paul when he comes to lecture at a course I organize at Cold Spring Harbor. And today we get to learn even more. I'm excited. So.
Thanks.
You're welcome.
Am I on? Can you hear me? All right, great. So yeah, first I just have to thank you, Sophia, and thank all the organizers. So Sue and Chris, and they've already started the timer. Okay, yeah, you know me well. All right, for the invitation. So first I just wanted to. So this is I am now a repeat speaker here. And so I was. How many of you were at were there for the Biocuration meeting in Japan in 2010? Yep. Okay. So you probably don't remember at all that I gave a talk there. Okay. This one will be just a little bit different. You know, believe it or not, we've accomplished a few things in the last 15 years. Okay. So I really appreciated this invitation. Okay, because you know how it is. You never really get a chance, I think, or you don't often get a chance because you're really just, day-to-day things that you have to do to keep large projects running. You don't often get a chance to step back and start to ask a few big questions. So I'm going to give a talk that has some parts that are pretty well, that I know pretty well, that I've talked about before. And if you've seen me talk, you'll recognize some of it. But I'm also going to speculate a little and use this opportunity to do a little bit of thinking outside the box and take or leave what I tell you. Okay, so the rough structure I'm going to talk about is I'm going to give you a little background for protein function prediction and annotation. Okay, and I'll explain sort of what is it. I'll try to explain why it's important. But I'll also try to give you some idea of the history and some key developments that have happened along the way. It should help give you some context for the talks in this session. If you know a lot about protein function prediction, you'll easily see what I'm doing. You might see a slightly different take on things you already know. But if you don't know, bear with me. So I know it's early. Okay, I'm going to ask people to actually, you're going to have to do some thinking probably through this, so just pay attention if you can. And then at the end, I'll talk to you about some recent work we've done, and then what I think maybe we can do to think about where AI can take us in the problem of function prediction. Okay, so what is function annotation? Okay, so I'm just going to step back and make sure we're all on the same page. It's really simple. I'm just trying to come up with some sort of description of the function of a biological molecule. Okay, and I'm not going to quibble with whether I'm talking about a protein function or a gene function, right? Because we all know the central dogma. Okay, so we've got a pretty deterministic process going from the gene to protein function. Okay, so it's often the proteins that are actually performing the functions, or in many cases, but not nearly as commonly, non-coding RNAs. So you may hear me switch between protein function, gene function. Don't worry too much about that, okay? All right. So why do we care? Okay, well, it's really that, we have lots and lots of genome information now. Okay, I mean, genomes, as you know, right? They're being sequenced all the time. Okay, so, but how is it that we're going to turn that information into actual biological knowledge? Well, we've got to we got to ultimately link the genome, right? The genes contain the code, right? How do we decode that, right? How do we understand? We basically go from the genes that we now know. We know that the full gene complements full genomes of huge number of organisms. But how do we link that to organism biology? How do we link it to diseases, ecology? Okay, well, the first step as we sort of come out from the genome is, what are these genes doing? Okay, And there's a really nice review. I am not going to ask you to look at this figure and try to understand it. Just to say, I think Chris Henry had a very nice paper on this. If you want to look at this in detail, sort of on how maybe computationally we can start to make that transition and start to understand phenotype from genotype, this is a great place to start. I just want to point out that it's going to start with assigning functions, okay? Functions to families, and you'll see, because lots of you have basically related genes in different organisms, and related genes, in fact, within the same organism, you often think of assigning function a sort of a family level. And Dan Haft will actually have a really nice talk on this later on in the session. So, okay, so I just want to again get our terminology down. Okay, there's function annotation and there's function prediction. Okay, and I would consider that annotation includes both capturing the knowledge we already have. Okay, so going into the literature, reading papers, or somehow doing it with NLP, whatever, trying to extract the knowledge that we have and capture that. Okay, so based on experiment. This, we know very little experimentally about almost all the genes from these organisms that have been sequenced so far. Okay, but we want to leverage that information, right, and use that information to help us predict functions. And this is the big problem, right? This is, at least statistically, I don't know what most of these genes are doing. This is what I want to try to do. I want to make inferences about a function that has not yet been experimentally determined. Okay, so what are what's one of the real roadblocks we have in trying to do this is that it's hard even to define what you mean by function. And I think I think everybody in this room has sort of grown up in the era, or most people in this room have grown up in the era of the gene ontology, and we sort of think, oh, it's out there. We know it. We kind of got an idea of how to describe function, but there was a time when that was not true. And so what sort of makes this challenging, and I'm going to walk you through a little bit of sort of how we've conceptualized as a scientific community, how we've conceptualized function through the years. So one of the things that makes it really challenging is just the sheer diversity of functions that genes have. Okay, and it's sort of the limit. There's something that's unique about every gene in every organism. Okay, and you basically got the snowflake situation, but it's not it turns out it's not infinite. Okay, and we can start to enumerate it, or at least enumerate pieces of it. Maybe combinations will be unique. So you really want to capture these extensive commonalities. Okay, and then the other problem with gene function is what is it? And I think there was a time, again, you'd read a paper and you'd say, well, I know it when I see it. That looks like a function description. Okay, and this is, again, it's not obvious at all what constitutes a function description. Okay, so how did we start as a biocuration community? Right, what was the early biocuration for gene function descriptions? And this is really where I think many of us, at least in this room, know the role that SwissProt played in this process. But Amos Barak was really a pioneer in this area. And you can start out by thinking about free text. Okay, I can name proteins. Okay. And we heard from Randy yesterday about all the issues with proteins and synonyms. You can only encode so much knowledge in a name, and we tend to want to do it, right? But this is really not a scalable solution. SwissProt solved this problem. At least the first iteration was, well, all right, let's do it with free text. I can write a description. Curated description, and this is done at all the model organism databases for a period of time. We were writing, in NCBI, they were writing curated descriptions. Okay, and this is a good way to think about it. There are pros and cons, right? Obviously, with free text, it's very flexible. You can capture anything you want. There's a con to that, right? It's not standardized, right? Are there ways that we should be thinking about this in a standard way? It's rich, all right? You can express just about anything, but that makes it complicated. So the next real advance, I'd say, in curating gene function descriptions was the notion of classification. And again, I would point to DisProt. The first real classification along these lines were the DisProt keywords, those of you who remember these. And just think of them as simple tags you can put onto proteins that now put them into categories that you can then look at and browse. And this is a really nice way of being able to handle the complexity. The enzyme classification, again, people are really quite familiar with, I think most people are familiar with the enzyme classification. It applies only to enzymes, so it's just a part of the functions that genes can have, but it's a very nice organization of how enzymes function. And so going sort of beyond that to a general purpose classifications that we're really trying to expand out to cover the wide range of concepts that you have to cover to really describe protein function. Monica Riley's work with GenProt EC, okay, these were purpose-built for individual genomes, capturing information in well-studied genomes. So EC, the GenProt EC was for E. coli, right? MIPS was developed for yeast, and fly function tree, you probably recognize, might be, anybody? Drosophila. Okay, so there are pros and cons with this too, right? It's standardized. This is fantastic, okay? But standardization doesn't come without a cost. Somebody's got to do a lot of work. There's a lot of work in just creating it, maintaining it, revising it. It's also rich. But to get that richness, you have to have a really large number of terms and relations between them. It gets big. Okay, so people probably are, has anybody heard of fly function tree? Yeah, okay. So you know where I'm going with this. All right, so the idea was that it basically, because of deep conservation of genes and functions among at least eukaryotes at the time, you could maybe think in terms of, all right, I could take this ontology that was really built, right, this classification that was built for fly genes, and I can start extending it so that it can be universally applicable. And this became what we all know today as the gene ontology. What did Go do that I think were really important advances? And again, these are things that maybe you don't think about. I certainly had never thought about before, that were really important advances, right? One of the things that I really want to focus on is that it really dared to define what we mean by function. Okay, and I just want to point out this little quote. Oops, where's my pointer? Actually, let me use this so I don't have, do I have a, okay, anyway, all right, sorry. Okay, so is this my, there we go. All right, so this quote is an actual quote from that first Go paper. And it's the vagueness of the term function that this paper tried to disentangle. It really tried to describe, tried to break it down into what do we really mean by function. And it resulted in what you now see today as these three orthogonal types of functional characteristics. Again, I'm not going to go into a lot of the details. Come see me if you want to hear about exactly what these mean. But basically, there are different levels of biological organization at which one can describe function. Another important advance is this graph structure. Okay, so it's not just a hierarchy. Okay, child terms can have multiple parentage because there are multiple ways to classify something, right? Or really multiple ways to think about its function. So, for example, I've got the insulin receptor, right? Insulin receptor activity. I can think of it as being part of a larger pathway. I can think of it as being a transmembrane receptor type tyrosine kinase. And I can also think of it as a hormone receptor, okay? These are all sort of different ways, different features, and really different characteristics of the function. of the insulin receptor. Okay, the other thing that allows me to do that's really important is that functions can be described with varying degrees of specificity and precision. This is really important because our knowledge is often incomplete. And this is another thing that's really important to be able to capture when you're describing function is I know it well, or I just know a few little things about it and I know it very vaguely. Okay, what else did it do? Well, by making this definition, it actually said, all right, now what do I do to make a gene description? I have some set now of functional characteristics, each of which is represented by a Go term. And that set together is going to be my description of what that gene does. Again, not obvious, but it's what came out of what came out of Go. We also defined, right, at the time, defined the function annotation process, right? It's the process of associating these terms with a gene. And then of course the important, I think one of the important advances that is really underutilized, I would say, by a lot of Go users, is that every one of these statements, it's really an assertion about something we know about function, is associated with evidence and allows us to distinguish between, is this experimental evidence or is it a prediction? If it's an experiment, what type of experiment? If it's a prediction, what method was used to make that prediction? Okay, and all of this is stored as metadata. Gene ontology got really big really fast. As I said, you get a large number of terms. I mean, just look at the number of Go terms here. Okay, we're talking each of this, I've just broken this down, not by total terms, but each of the separate, right, molecular function, biological process, cellular component, 3 different types of function descriptions, right, function characteristics. And you can see, I mean, I'm talking, you know, we're talking reaching biological process, reaching almost 30,000 terms at its peak. The ontology is really big. But if you kind of break it down into periods of time, there's what I sort of call the organic growth phase. And really at this point, we've got a lot of different groups, annotation groups, annotating gene functions for their organisms. They're requesting concepts to be added. They're being added at a very, very fast rate to cover the descriptions that are in the literature. Okay, but then you can see after a period of time, we start to saturate. So remember, this is just the total terms. There's lots and lots of, you know, adding of terms and deleting of terms during this whole process. And so at some point, we start deleting or, you know, obsoleting terms faster than we're adding them. Okay, so this is hiding a lot of complexity. And right now we're at about 300 terms added a year, 900 removed per year. Okay, total across the 3. So what is driving this? Again, there are still requests from annotators, but they're much less frequent because we've captured a lot of the concepts in the literature. But what we're doing more and more of is now taking that global view. What is the structure that's been created by this organic growth process? Going back and working with experts and having them help us revise and make sure each branch of the ontology, which represents different areas of biological expertise, and you can see some examples up here, make sure that it meets, that it matches the current way of conceptualizing function in those fields. Okay, so there's a lot of revision going on. I need to add also, of course, that some functional characteristics are not covered by Go terms at all. And so if we're thinking about how ultimately we need to be describing function, we're going to have to go beyond what's currently in the gene ontology. So some types of molecular specificity are not covered. And again, I can get into details with people who are really interested in what we aren't covering. Steps, individual steps in pathways are not covered, okay? But they are captured by other resources and actually by Go causal activity modeling, the GoCam framework that I won't talk about today, but I'm happy to talk to anybody about it online. But you know about this. There was a great talk on plant reactome yesterday, so you're familiar with basically pathway resources. I've named a few of the more popular ones here, and that's where you can get this additional information. Okay. Go primary annotation process has been really effective. And by primary, I mean extracting Go annotations from the literature based on experimental results. You can see just the total number of publications that have supported annotations. It's huge at this point, resulting in close to on the order of 1 million annotations, as well as these are Go terms, and these are protein-protein interactions, which are these protein binding annotations that you probably hear about. All right, so I'm going to switch gears now. Hopefully we're all on the same page about what function is, what the gene ontology has done to structure knowledge, and to extract information from the literature. What can we do to use that information now in prediction? All right, and I'll just give you the way that the sort of basic paradigm for function prediction. It's often called annotation transfer. Okay, so just consider this simple case. Okay, I've got a human protein that's unannotated. Okay, I don't know what it does. Nobody's done an experiment yet, or at least it's not in the Go database. Okay, somebody has done an experiment on a yeast protein. They found that it has this particular enzymatic activity. Let's say I would like to be able to annotate this human protein, and I find that I can establish some sort of relationship between the human protein and the yeast protein. I'll get into what those relationships can be in a minute. Then, given that relationship, maybe under the right circumstances, I can say, all right, I'm going to make a prediction now. I'm going to transfer the information that I had on one protein, right, from experiment to a protein that I don't have any information about. The problem, of course, with this is, okay, which characteristics can I transfer? Can I just say, wow, everything I know about it, I can just pour it over to this other protein? Turns out, of course, obviously, it's not as simple as that, or we'd be out of a job in protein function prediction. So, of course, it depends on the relationship. What's the relationship that you established? What's the nature of the characteristic that you're trying to transfer? And so, for things like pathways and shared processes that genes perform together in a group, there are actually some signals that you can use from various different other data analyses that can help give you a hint at that these genes may be working together. And so some of these, I've listed some of these again. I think they're probably getting into too much detail is gonna be a little too much. But again, I just want you to know there's a whole literature behind this and some really clever work that's gone on in this field. So, I will just give some of the basic ideas. Okay, the Rosetta Stone method, this was published in Science, at the turn of the millennium. It's amazing. From Ed Marcotte and David Eisenberg. And this is basically based on the idea that I can actually, if I survey a lot of microbial genomes, I find that occasionally, but really rarely, I find two distinct proteins, as they're found as distinct genes across many, many genomes, but in some genomes, they're fused into a single protein product. Good hint that they're probably working together, okay, because they're guaranteed to be co-expressed if they're covalently linked to each other, right, in one polypeptide chain. Phylogenetic profiles is another method where if I look across a lot of genomes, I can see that some genes tend to get lost at the same point independently in many different lineages over evolution, or they're gained together. They all appeared at the same time. These are hints that these might actually be working together as a functional module. So this is just to give you an idea of the kinds of clever work that people have done. Okay, so of course I'm going to say that by far the most successful, certainly the most widely used, is inference from homology. Okay, and this is partly because it's general purpose. You can predict any, at least in principle, right? Any feature, any type of functional characteristic. But of course, as I said, the details matter. Depending on the relationship, I may be able to say more or less about the function characteristics that I think I can transfer. Who in this room has ever done a blast search? All right. Okay, that's good. All right, so you know about how this works. maybe you've thought about it as homology searching. Maybe you think of it as similarity searching. But either way, okay, and I'll get into sort of how to conceptualize BLAST searches. But this is really widely used, right? I've established homology via homology search. And then what do I transfer? Well, actually, Bill Pearson, who sort of I think of as the real guru in this field, would say, yeah, it's case by case. But if you have a really great E value, like, better than this one, I think he was saying, 10 to the minus 60. So in other words, so one in 10 to the minus 60 chance, 1 in 10 to the 60 chance that this matches by chance. It's homologous for sure, but it's also so closely related that it's probably doing the same thing. But this even is not that close. So in that sort of cutoff for I can definitely transfer everything I know from one protein to another based on BLAST, this would not meet that criterion. Okay, so it's really case by case, and we can't afford as biocurators to review every possible pair, right? I've got a pairwise problem. So it's not just that I've got, n annotated genes, I've got n squared relationships. So it starts getting really, really nasty. Okay, so there are other ways to get around this orthology. I'm not going to give a lecture on what orthology is, but I do have a real soapbox I love to stand on for that one. So if you want to hear more than you can handle about orthology, please come see me. Okay, but again, the issue with orthologs is that, of course, you can see that if let's say I'm trying to look for orthologs with this, let's say I'm looking for orthologs in mammals of a gene in Xenbase, then I may find that there has been duplication after the divergence of these genes, and I've got multiple orthologs. It makes things complicated. Okay, and so when I don't have post speciation duplication events, so gene duplication, of course, right, is how many new gene functions come into existence. And when gene duplication happens, you got to be careful about what you transfer. But if gene duplication hadn't happened, I'd have one-to-one orthologs. I can transfer reliably many characteristics, okay? But it quickly gets more complicated. And then family-wise, okay? The family-wise approach, and again, you'll hear about this from Dan later on in this session. But again, the important thing to know is that you really then you try to say, what can I say about a whole family? And then I can just establish homology relationship to an entire family. And anything that I think is in common to the whole family is something that I can infer for any of its members. Okay. And Interpro is a really highly used resource. How many people use Interpro? Is that also okay? So we also know this. All right, great. And what's so great about Interpro and the family-based approach is it starts to be tractable now for biocuration. I think this is why you use it. It's a good way to at least start getting down into the details of function that can be curated, and you can get reliable annotations. And disclaimer, of course, I'm in the Interpro consortium, so I'm gonna sell it to you. All right, but I really think actually it's a great resource. And it's based on this idea of families or breaking up families into smaller families for which you can reliably assign function, at least you think you can. And so, as I said, Dan is going to be talking later on, so you'll hear a lot about this. But there are a number of data resources here, and then Interpro actually has, this is a really great publication on how, if you want to know actually exactly how they assign, what the process is for assigning functions to entire families. It's well thought through. All right, now I'm going to get into, let's think about what we mean by homology. So if you were kind of glazing over a minute ago, I know this is a lot of information in a short amount of time. I apologize, it's early. I wish we could take a coffee break before this next section, but I really want you to think about this because this is really important. Okay. All right, we're all awake again? Okay, great. All right. I want you to think about what it means to do an annotation transfer. Okay, what does it actually mean? What am I doing? What's my model? And I actually have a model of a biological model of the meaning of that. Okay, and I'll try to show you what I mean. All right, so again, this is our BLAST results. Okay, so I've got my query as a human protein. Okay, I don't know what it does. Okay, my subject that I found in my BLAST search is a yeast protein that has been annotated with experimental information about its function. Okay, and what I'd like to do is I want to transfer that to the query. So I want to say something about the human protein based on what I know about the yeast protein. What does it mean if I do that? Okay, here's what it means. Oops. Here's what it means. I actually have an implicit evolutionary model behind what I'm doing. Again, I don't know. Have people thought about this when you do BLAST? Yeah, good. You should. Okay, I've got a yeast protein. It's annotated. I've got a human protein. What I'm saying is I just did a BLAST search. What the BLAST search does is it does this. It says that there was a protein in the last common ancestor of human and yeast over 1 billion years ago. There was a protein. And from that protein descended these 2 extant proteins. That's your model so far. Now, that's what BLAST is telling you, right? What does it tell you that I want to transfer that annotation? Remember, these are very difficult. These are very complex functions. It takes a lot to evolve them. They are very unlikely to have evolved twice. And so what it's saying is that it's now making a statement about this ancient protein that doesn't exist anymore. And it's saying it was already a methylene tetrahydrofolate reductase. It was able to catalyze that reaction, okay? And then it says, then my inference then says, and that was also, so not only do I say something about the ancestor, but I say something in an unbroken chain that was passed down from organism to its progeny for 1 billion years to get to the human protein. That's what I'm doing when I do BLAST. Okay, there's a model. Okay, and you can see now that this is starting to look like what you will recognize as a branching point in evolution and branches, right? So there's independent sequence evolution along each of these branches. They're separate now. They're not the same. Up till this point in evolution, right, there was no distinction between ultimately what would become the human protein and the yeast protein. It's all just up to this ancestor. But then at that point, you get divergence of 2 lineages. They go on to independently change in sequence, which is why you see that this isn't a perfect match, but they don't change in function. Okay. So then, like I said, for representing evolution, who do you think came up with the representation of evolutionary events that we still use today? What's that? Yep. Bingo. There was, so when The Origin of Species came out in 1859, there was exactly one figure in it. Because figures were really expensive. You had to make plates and do the whole, anyway, there's exactly one figure, and I'm going to show it to you in case you haven't seen it. That's the figure. It's a tree. It's so the basic idea is really simple, right? The basic idea is that I have things that are in common. These are ancestors. They, at some point, they become separated. They independently change. That process happens over and over again, right? You get, basically you start out with 2 things that are exactly the same, but once they're evolving separately, they're going to evolve in different directions. They're going to accumulate differences, and that looks like a tree. And we still use this conception today. So, again, if you sort of want to understand all of the, understand evolution, learn to think in trees. So I'm going to give you a bunch of trees. Maybe you are all familiar with trees, but there was a point I had to learn trees, and it's kind of like a religious experience, honestly. Okay. So the annotation of families then also implies a model of evolution. What's our model of evolution in this case? Okay. The model is I have consistent functions that are annotated all family members. Inconsistent functions are ignored. Okay. And so imagine I've got a bunch of sequences, some are labeled, right? With each of these colors is a different function. And I'm just going to say, all right, give me the things that look consistent across the entire family. And what does that mean in evolution? It just means that, I've got some sort of tree that relates all these sequences to each other. I overlay them on a tree and I see that, yeah, these look like they're in common. And what I can say now is, I can say that this function was present in that common ancestor, just like I did in the case of human and yeast, okay? And then that was then propagated to its progeny. And now look at me, I've got an annotation tool. But what if the inconsistent functions are really common? Well, you can see already from the tree, right? I can start to do things like, oh, well, this, maybe this function arose here in the tree, and that would be consistent with what I see. Maybe the red function arose here, and I can actually start to go down and start to say more and more specific things as I get closer and closer to the leaves of the tree. And that's, in a nutshell, what we did. Okay, we basically built an entire infrastructure around treating the function annotation problem like an evolutionary reconstruction problem. In other words, going from these implicit models of evolution to explicit ones, where we capture the entire model. Okay, and I don't know if people are paying attention to the literature in the last couple of weeks, but we published a paper. We had a public paper in Nature from the Gene Ontology Consortium about applying this approach at massive scale. So we're talking about doing this for over 6,000 gene trees that together cover almost all human protein coding genes. All right, and again, I'm not going to walk through this. You can read the paper. But what I want you to see here for the purposes of this talk is that biocuration was essential for us to do this. All right, and it really happened in two different kinds of biocuration were employed. One, as I said, I've already talked to you about primary Go annotation, right? You go to the literature, you find Go terms that you can be that describe that functional characteristic. You support it with the evidence from that paper, giving us a massive corpus, as I said, of information. But what I haven't told you about, and what I've sort of hinted at, is that we actually then went and needed to build these evolutionary models. And we also did this with biocuration. Lots of reasons for it. I'm happy to spend time with people about explaining why. Bottom line is, it's a really complicated problem. And at the time, there wasn't a computer that would do it. Who knows, maybe we'll figure out how to get AI to do it. We certainly have a big training set now. Okay, so we created evolutionary models at a massive scale, used that to infer protein functions or gene functions where they weren't known, and came up with a fairly complete compendium of human gene functions that we think is quite reliable. But it's not complete, and I'll get there. Oh yeah, the other thing I really wanted to point out is, the biocurators. I mean, I really want to acknowledge this. These are the biocurators. They're the first authors in the paper. Yes, you recognize the names. Okay, we really had some superstars working on this project, and that was a huge measure of our success, and I really want to give that shout out. So I'm gonna, in the interest of time, I'm gonna skip this because I really want to get. I think you get the idea. Okay, I can drive this into the ground. Okay, but I want to explain what the important advances were in this approach. Okay, it's basically that it's an integration problem now. I've got all these annotations, right, as I said, on the leaves of this tree. And I can actually now think about this evolutionary framework as a way to integrate all of the information that I've got from various different organisms, different related genes, different genes in the same family that may be from the same. So I have, both paralogs and orthologs, basically. I have genes within related genes within genomes, and genes that have diverged via speciation. So the other thing that I think it does that's really important and that's new is this idea of selecting independent functional characteristics. And I'll give you an example of this because it's really important. each characteristic may have a distinct evolutionary history, but the take home is that just because you have a distinct go term doesn't mean you have a distinct functional characteristic. Okay, can we wrap our heads around that one? That's the main point. And I'll just give you a quick example. Okay, quick example is, let's look at this one. Okay, so quick example is, in red are the chosen, the selected during this curated process, what the curator chose to model, okay? And for example, they felt, all right, I've already called it a cytoplasmic pattern recognition receptor, okay? So, but I want to specifically say it's involved in the antiviral response, so I need another term to do that, okay? But once I've chosen these two, it's like, well, all right, then that now explains why it's been observed to be involved in negative regulation or positive regulation of various different cytokines. It's upstream of cytokine activation, but cytokine activation is not what it's doing directly. It's really part of a pathway, okay? So we're trying to capture this modularity of biology, which is the function does not comprise absolutely everything that happens downstream. Okay, and that's what's important about this selection. You can see we're selecting, in some cases, a pretty small subset. So the idea is kind of like the old parable, okay, which basically to me in this case is meaning that when I'm looking from just a point of view of a paper, I'm making an experimental annotation, I'm looking at one paper, I'm describing that exact feature of function that's described in that paper. I'm not taking the global view. And when I bring all the information in, I'm able to make those global decisions. Okay, and this is if I only show one slide, which might be the case, if I only show one slide, I want to make a call out to all of you in the audience, because this might be, one of the amazing things we found when we took this massive corpus of Go annotations that have been made over many years and we integrated it all, we are still missing known information. So I'd love it if you went to our website and, and we can get as many people as we can and to take a look. And if you have a favorite gene, something that you know about, or you know about its relative in another organism, and we're missing information, it would be great to be able to add those experimental annotations to the Go knowledge base. Because remember, we only made inference based on experimental annotations. The experimental annotation was missing in that family. We couldn't model it. Okay. All right. Great. So in my remaining time, I will talk about, really briefly, apparently, but that's about as much as you'll get from me, what it'll take to solve the protein function prediction problem. And here's the true answer. All right. I mean, let's be honest. Okay. But, you invited me. So I do want to speculate. And one of the things so I don't know if anybody saw this paper from Christoph Decimo and myself, but we used basically structure prediction as a great example of what AI can do. Okay, the protein folding problem, or at least the protein structure prediction problem, has been largely solved. It's been solved by AI. Okay, so can we use that as an analogy for what we're going to try to do with function? Okay, and it's not, and so you can think of this as a black box solution, which of course AlphaFold 2, which was the program that originally, or the AI that solved this problem, it's not a black box. Okay, there's actually a design to it, and that design relied on a lot of previous knowledge. And I'm not going to get into all of the previous knowledge, but just suffice it to say this paper is fantastic, from 1994, from Chris Sander. And the idea was already out there of the basic structure that you would want in a solution to the protein folding problem. But for many different reasons, we didn't have a computational means of performing all of this because it's fuzzy information. And you have to be able to refine your fuzziness, which is something this turned out to be a really great problem for AI. But again, I can go into a lot of details with people if you care, but there are sort of two essential pieces right? Which is, we already knew if you take it and think of it as a problem that you want to solve for many different proteins simultaneously, because we know that if the sequence is similar, the structure will be essentially the same, okay, to a good degree of approximation, okay? And it's all about doing your first step as approximate. And that there is information about, in these alignments, about 3D proximity. And this was already known. But how do you turn that into a structure that's a hard problem, and you have to encode all sorts of things about chemistry? And again, I'm not going to get into the details, but you can give information. There's lots of information about chemistry that can be used in this problem. And the other thing that you have to do to be able to have AI solve this problem is you need extensive training sets. Okay, and these tend to be models and not raw data. Okay, and I'll talk to I won't talk about that in this talk, but ask me about it. Okay, and then at the end, and Iddo is going to be able to shed some light on this in protein function prediction, you have to have a clear target for both training and evaluation. What is it you're trying to predict exactly? Okay. Again, don't read this slide, but I just want you to take home that there are a lot of analogies that we can use to port from structure prediction to function prediction. And in red are the things where maybe the analogy is not so great. And we're going to have to think outside the box and have a different structure maybe to an AI solution. Okay, and we're going to have to handle things like I was telling you, that I can no longer assume that it's related and has the same function like I could with structure. Okay, and it gets back to my original slides on the complexity of representing function. Okay, and again, I just want to point out really quickly, this is being done as we speak. People are already starting to figure out how I can use information from structure to feed that back in, use information, at least an approximation of evolution from sequence embeddings. There's already a lot of work in this field. Okay, so the last slide I wanted to have here is the real challenge that we still face in all of this, is that modeling function is still, I think, more challenging than structure. And I'm gonna suggest that maybe this means that it's going to be more challenging to train, to design an AI, train it, and evaluate it. And it's a simple argument, okay? For structure, I'm modeling a 3D object. We all know how to represent a 3D object. This is what was so beautiful about what PDB did was, you don't provide raw electron density. You basically say, here are my inferred atomic coordinates in the Cartesian coordinates. Everybody gets it, right? And you're modeling a 3D physical object. You're also, the unit of publication is a structure, okay? And that also means you can evaluate publication by publication. Did I get this right? Okay, and it makes evaluation much easier. This is not true for function. We've gone through why. Okay, function's more complicated, and as you saw, bits and pieces of function information come from distinct publications. Okay, it makes this entire process much more challenging. But I think, of course, biocuration can help to solve this problem. So what can we all do? We can define the problem and what a solution looks like. And we can provide those knowledge sets. Again, I wouldn't think of them as data sets. They are really sets of knowledge. And this is why it takes biocuration for AI training and evaluation. Okay, and so my last slide, of course, is my pitch that, I really think that this community, should think of itself, and maybe you already do, as playing an essential role in the scientific process. You're democratizing knowledge by making it broadly accessible, and it's only really accessible. The audience gets more and more accessible as you progress from data to knowledge. The more it looks like knowledge, the more people can use it, the more different machines can use it, the more AI can use it. Okay, so there's my summary. Okay, function prediction is still an outstanding problem. You can use large-scale evolutionary modeling, at least to take some really large steps in this area. And that all of you can help in modeling this functional knowledge. You know, we encourage feedback on the ontology and the annotations. And of course, my obligatory cast of thousands, because this work is not my own. Anyway, thank you.
This is kind of like a COVID thing, right? We all took a little break for a while. Now you're going to have to reboot.
Hey, Paul. So again, as always, fantastic talk. There was one area I didn't hear you address, and that has to do with the concept of reversible reactions. And I'll give you like 3 levels of examples here. So there are a number of kinase phosphatase pairs, which are essentially the same protein, but homologous in different organisms or whatever, that catalyze the same reaction in opposite directions. There are also, when you think about it on the pathway level, there are pathways that run one way in one organism and the opposite way in the other. I'm thinking of photosynthesis versus glucose metabolism. And at the developmental level, an example would be the development of an eyeball, which takes light stimulation, converts it to an electrical signal to the brain, versus the development of an aquatic organ known as a photophore, which takes an electrical signal from the brain and converts it to a chemical signal, which produces light, which is sent out via a lens. Is there any way that you can figure out how to dissect the directionality of the reactions using these techniques that you described?
Yeah, wow, that's a great question. Thanks, Mike. I also just want to give Mike a shout out. So you may have remembered from my 2010 talk, Mike was our first guinea pig curator. I really feel sorry for you, actually, for building these evolutionary models. And you had to do a huge amount of very painful work while we figured out exactly how to create a whole new SOP for a whole new kind of curation. So just wanted to point that out. So yeah, thanks for the question. So again, I went through a huge number of slides really fast, and maybe not quite fast enough. But one of the slides I had was about sort of what is missing from the Go terms, and a lot of these are in. A lot of the questions you asked and the additional information you'd want to see, things like, yes, it's reversible, but in this context, here's how it runs. These are actually causal relations and a flow that we try to capture in GoCams. And so these are extensions to Go that add on top of the basics that you already have in the Go term. So a Go term is designed to let's take the simplest case, okay, the reversible enzyme. If it's reversible, right, in principle, we will have a Go term that covers that reversible reaction. And you will then, once it's embedded in a context, in a specific biological context, so particular organism, maybe a particular time, maybe under particular conditions, then you can actually specify what the flow looks like under those conditions. So in other words, it requires a lot of extra metadata, which will reside outside of the Go in other ontologies that we then link together into a more complete model. Does that answer the? Great. Okay.
Yeah. Hi.
Hi.
Excellent lecture. So I have a question regarding experimental. So when we conduct experiments, wet lab experiments, mutant analysis or transgenic analysis or whatever, we end up finding that there are several genes which are probably not annotated as having any function, or they have a domain of unknown function. So while we can figure out the biological process in which they probably are involved because they are either stress regulator or something of that sort, But when we try to pinpoint to a molecular function, we are totally in a dark how to conduct and which experiment to conduct to figure out the molecular function. So a comment from you would be great. How to go about this?
Ah, you mean, oh, advice on how to, yeah,
Yeah.
that's, yeah. So yeah, that's a really interesting question. That's a great question. I don't think I've gotten that question before. So, now just thinking, so thinking a little outside the box, what we have a lot of are, for many of our Go terms, we have an enormous amount of documentation that I did not talk about. So, again, I talked about the size of the ontology, but you can imagine keeping our annotations consistent, making sure everybody, all the annotators across the consortium are making consistent annotations. We have a lot of guidelines, and those guidelines often refer to the kinds of experiments that would have to be done to establish the presence of a particular functional characteristic.
Okay.
So again, it's this may be helpful if you're looking for, is this a transcription factor, for example? What would I have to show?
Right.
And so, but again, doing the reverse, I think a lot of people are doing these high throughput phenotyping experiments now, where you can actually, basically see, look at, the phenotype under various different conditions. And then it's not just one condition, as you said, right? You may have
Right.
response
Yeah.
in one condition. When you have a large number of conditions, you can often start to narrow down the possible functions.
Actually,
Yeah.
that's a good point. So actually, when we look at those proteins, some of them even don't have any recognized protein domain. So I can't even figure it out whether it's a transcription factor. I don't have any leads on that. And there are several number of proteins of that sort where either it is domain of unknown function or there is no functional domain which is known.
Right.
So maybe I can get back and we can have a discussion.
Sure. Yeah.
Thank you.
Thanks.
Yeah.
Thanks. Good question.
Thank you so much. Is this one on? Yep.
Yeah.
Thank you so much, Paul. We're going to be moving ahead with our schedule just to stay on schedule. So I encourage all of you to harass Paul as you see him about the meeting with questions and again, thank you very much.
Thanks.
On. Hi. Yeah. Okay, so talk to follow. I am Victoria, and I am a postdoc, and I work as a lead curator for DisProt database. So I'm going to tell you a bit about our resource and my work. Let's see if this works. Okay, so yeah, DisProt is the database of intrinsically disordered proteins or regions. These are proteins that intrinsically lack a fixed 3D structure. So they are very flexible. They can have regions within them that has really flexible characteristics. So what we do is that we capture these proteins and we curate them. This is a database that's based on manual curation done by community curators and professional curators as myself. And in the database, we collect the information that is experimentally validated and captures the features related to the structural aspect and also the functional aspect of these special proteins. So this is what of our entries look like. So what we do is that we create a dedicated entry based on the UniProt ID of these proteins. And we have some general information about the protein, and then we have all the pieces of information or annotations there that again are related to structural states, aka disease disorder, transitions that could happen to a more ordered state or structural states, and also functions that these regions can be involved in. So for all of the different types of annotations that we do, the pieces that we curate are the same. So for example, this is an example of one of our functional annotations. You can find, and this is the information that our curators add. We have the details from the publication, some statements or snippets from the publication, what is the thing that we are annotating or informing about. So in this case, it's a function, and we specify the function using gene ontology terms. We specify the fragment or the peptide, the region, I'm sorry, of the protein that we are describing, the method that was used for the assessment using eco terms, and when we have the possibility, we also add some experimental details like phosphorylations, the presence of other proteins, and stuff like that. So DisProt has been growing steadily since its beginnings, both in the number of entries or proteins that we have and the amount of annotations or informations that we have about them. But this is not all. That's more than meets the eye. Sometimes when we are applying for funding or trying to get across the efforts that we do in a database, we tend to only showcase the size and how it's growing in size. But there's more to it. And I get that it's difficult to measure, but there's also the quality improvements and the updates that we do in the databases. So just to show an example of what I'm trying to say is that in DisProt we have over 12,000 annotations, and now I'm very happy to say that more than 50% of those are validated. This means this is related to the revision process that we have in place. So when a biocurator, community curator, whoever adds a piece of information, do an annotation in DisProt, it goes to us reviewers, and we kind of do a double check on the information. So if the annotation is lacking or could be improved in some manner way, we provide feedbacks and request changes from our curators. And once that annotation is perfectly curated and annotated, it gets validated and it gets ready for the releases that we do each 6 months. So what I'm trying to say is that now 50% of the amount of the annotations and information is double checked and validated. So we have enhanced the confidence in the data's accuracy. And the fact that we have 52% means DisProt has been here for 2016. We started to do this revision process 2022 actually. So in these 3 years, what we have been doing is not only validating and double checking the new information that's been added in DisProt, but we are going back and trying to make sure that the annotations that are already there meets today's criteria as well. So we are not only validating, we are also updating things that are there. And this is a difficult thing to show how much annotations have been changing, but updates are important. And this is not the only trigger of updates in our database. We have some external triggers of updates. These are the more common challenges that we face. Firstly, UniProt updates. We relate to UniProt IDs. So every time UniProt merge entries, obsolete, change the sequence of the peptides or the proteins, we need to apply or to see how those changes also affect our database. Another thing that happened is gene ontology updates. So since we annotate functions using gene ontology terms, each and every time the branches change, evolve, we have to do it as well. So we need to go back to our annotations and make sure how we are aligning with these changes. We also have some internal triggers. We do these things to ourselves in a way. So we have our own ontologies that are also evolving and changing. And so we also need to double check and make sure that our annotations align to these changes that we are proposing to do ourselves. And of course, we are also trying to be aware of how structural biology has been evolving, all the predictions and all the different tools that we have. So we are also adopting new eco terms that relates or is the curation process for us or allow us to assess or annotate different things. So this is a lot of work because we are not only considering how we need to keep growing in size, but we also need to keep maintaining the quality of the database, doing the updates and doing a lot of things. So how do we do it? We are trying to I'm sorry. We are trying to also ease a bit of the load by relying on our community curators, and we try to support them as much as we can by a variety of different training and courses. So we have some online training courses that we do regularly, some in-person workshops whenever we can. We do also in-person workshops. We have been doing we've done a survey last year to see what our community creators needed from us or the challenges area. So we come up also with some trainings regarding some experimental techniques related to the assessment of IDPs. And we have some e-learning courses in the Elixir platform in Spanish and in English for them. Another way that we support community curators is by crediting them. We rely on Biocuration for that, which is the platform for crediting and acknowledging scientific contributions. Biocuration, what it does, it gives you like a score for the activities that you perform in this database to our curators. So in that sense, they earn badges and medals when they achieve different milestones. Biocuration also sends this participation to the curators' ORCID ID. And we, on the side of DisProt, we also acknowledge our community curators by in the co-author of our publications that we do in nucleic acid research every 2 years. So our next publication is coming up mid this year, probably September. And if you are interested in seeing other kinds of updates that we have been doing in the database, I think you might be interested in following this next publication of ours. And with that, I'm very happy to say that I'm okay with time. I'd like to thank you all for your attention. I'm very happy to be here with you all and meet you all in person. And this is my group. And of course, a shout out to our community curators. Thank you.
Okay, we have time for 2 questions. We have a mic runner.
I just have a question about on your website for like the repeated annotations. Is there something like a sort of a mark or something, information for the viewer that says this has been reviewed and the date?
Yeah, if I'm not sure. Yeah, if you check, yeah, for example, you have the annotations, you have the curator name, and also the validated by tag and the name of our senior curator that reviewed the annotation. So you can see that it's double check. Right now, we are not releasing anything just from those that are reviewers themselves, but we are not releasing any kind of annotation that is not validated. But you can find some of our older annotations without the tag.
Other questions?
Thank you. That was great. I think you probably know what I'm going to ask about. So yeah, so I know you you added a lot of you helped us actually add a lot of Go terms to gene ontology to handle disordered protein functions. Can you talk a little bit about your experience working with us, and what did we do well, and what could we have done better in trying to work with you to incorporate the terms?
I love that question. And I would love to talk about more in the gene ontology meeting in the next month. Well, I think the challenge you have is really, really difficult. So I think everything you're doing is great because the amount of work is difficult to handle. For us, particularly in our database, we have something that I always try to talk as a philosophical distinction. We are defining regions. You are defining genes, actually, and proteins in the extent. So there are some concepts or some things that I'm looking at the publication. I'm trying to say, is what I'm trying to say. Does this convey the idea? So for us, the challenge is to understand how to annotate this, that it all relates to a region and not to the whole protein. But we can talking about this because I have a lot of things to say about this, but I don't want to bore the rest of them.
Okay, thank you so much. Let's
Thank you.
thank one more time. And Iddo, you are on scale for protein function annotation.
All right, thanks. How many here were at ISMB in Montreal? Okay, this is going to be very familiar. So Clara, who is PEDGE student in Northeastern, gave a really nice version of this talk. I'm going to give a very compressed and not as good as hers version of this talk about crowdsourcing critical assessment of protein function annotation and algorithms. So I'd like to thank the organizers for a fantastic meeting and, you know, the free espresso machine and beautiful weather, neither of which was probably that cheap. And I would especially like to thank Paul for doing a lot of the heavy lifting that is required to explain, you know, protein function prediction versus annotation and all that. I completely agree with them. So basically, this is what protein function prediction can be viewed as when reduced to something computational. We have the protein sequence. We can take other parameters if we can find them, such as the structure if we have it, protein-protein interaction data, expression, literature mining, various other things. And from all of these, try to predict a subgraph which describes the function of the protein, one or more subgraphs from the gene ontology. That is how we view computationally the idea of the protein function prediction problem. So of course you can say, okay, this is not, Go is not the end all and be all of functions. There are phenotypes, there are other things. Right now, we actually have been using other ontologies, but the general idea is taking this input space and mapping it to one or more subgraphs or subdags, Go is a directed acyclic graph or a DAG, that describe the function. And that is a pretty decent, I think, representation of a computational way to predict function. So Go is huge. These are basically a summaries of the graphs of molecular function, biological process, cellular component that Paul talked about. So it's a very sparse, what we call a very sparse kind of language problem to take all this input space and find all those the exact nodes that are required to describe the function of the protein. It's a very complicated problem. And Paul talked about what will it take to get us to the AlphaFold moment? What will it take to get us to solve the protein function prediction problem? And he said he didn't know. I don't know why he doesn't know. I know why I don't know. And the reason I don't know exactly is that I don't really understand. You know, it's not one problem. It's a set of problems, or rather kind of a nebulous cloud of problems. But one thing I know is that to get closer to it, to get to some at least some practical solution, like we got with AlphaFold. AlphaFold did not exactly solve the protein structure folding problem. It kind of solved it more than solved it really. But to get to that point where we have something practical, like Nirav said, we have to go together to go far. So we have to have a bunch of communities. We have to have people from all the communities that are kind of stakeholders in this to work on this. And that is especially important in this community. Biocurators play a really important role, as we will see. And the other couple of two communities that we might talk about are, of course, bioinformaticians, experimental biologists, and the AI community at large. And we'll see how we try to bring these all together. So the way we try to bring this all together is by running this competition called CAFA, Critical Assessment of Function Annotation. It's a little bit of a misnomer. Paul talked about the differences between prediction and annotation. We're really trying to do critical assessment of function prediction. We call it annotation because saying CAF is really not very, you know, it's hard to say. CAFA sounds somewhat better. But really, this is kind of both. And this is what we call a prospective challenge. And here is how it works. We start here at a certain timeline where we give the people to predict the function, we give them a set of sequences, a very large set of sequences, more than 100,000 sequences. Say, predict on those, predict a function, find the Go terms that are attached to these sequences. Some of this is easy, some of this may be already solved, some of this is hard, some of this is impossible. And over a period of time, they do that. And then we tell them, okay, stop predicting, that's it, no more predictions. We save their predictions in some repository, and then we wait basically for the various database that annotate function to grow. So some of the proteins that didn't have a function here will have a function here. Well, they had a function, but not an annotated one. We didn't have Go terms associated. That's what I meant by don't have a function. And then finally, we run some metrics to assess how well those predictors mapped the Go terms to the protein sequences. And then we all get together, usually at ISMB, and everybody, talks about what they did, and it's a lot of fun. So this was the idea for CAFA1, which we started in 2010. And this is the timeline for the prediction phase and the annotation growth phase. Then in 2013, we did CAFA2. And then we did CAFA3. And then we did CAFA4. The annotation growth phase took a longer time. There was a little pandemic going on here. Pedja moved to another university. Various graduate students were really freaking out and stuff like that. So this paper is still in preparation, but you can read the other ones here. And over the past 14 years, the history of CAFA was kind of a history of growth. We had 30 teams competing in CAFA1, then in CAFA2 we had more. CAFA3 and CAFA4 kind of plateaued a little bit, but things weren't, we were working together. We were working with biocurators to give us data sets. We were working with bioinformaticians to do the predictions. We were working with experimentalists to give us data set. That was a lot of fun working with experimentalists, but we were disconnected from a really large community that in the machine learning field, all those people that do all those large language models and everything, and all the cutting edge stuff in AI and ML, we weren't talking with them as much as we should. And we started reaching out and talking. And finally, in a couple of years ago, we hit upon a great solution. And we were approached by Kaggle, a large data repository, a large site for data challenges and repositories. And they offered us to run CAFA on Kaggle. Kaggle has 2 million subscribers, most of them all of them from the machine learning community. Not a lot of them bioinformaticians, a small fraction, but they were really excited about this problem once we described it, how it is. And they said that, yeah, we will give you even cash prizes for the top 6. And that gave a huge incentive. And that really gave a great incentive. So we gave an evaluation set, a training set, leaderboard set, because the way CAFA runs is that they have this leaderboard and people submit predictions, and then they go up and down the leaderboard daily or hourly, really. And they can see how they're doing. And that's really cool. And finally, we had an evaluation set accumulated over time, like we usually have in CAFA. So we assessed everything a few months later. It's the nice thing about Kaggle, they have this ongoing feedback. So you have the leaderboard, you have these various teams. This is basically first to last. The top 6 are the ones who will get the prizes. So they're kind of marked here in green. You can see that this changes over time. People go up and down the leaderboard, and they communicate a lot. That is a great thing. This is a community. They talk with each other a lot. So, oops, sorry. So they do a lot of data sharing, code. They share their code. You're not allowed to share stuff offline at Kaggle. So everything goes online. These are people competing for quite substantial sums of money. They're still giving each other a leg up. They were organizing webinars, really nice webinars, explaining the basic of how we can apply various methods. So this is really great. And if you have an error, we had an error in one of our metric computations. This was reported by the participants. This went to the public evaluation code, was updated, and it worked. So that was great. We had participation. As you can see, participation really jumped from 70 in CAFA 4 to 1625 teams in CAFA 5. That was almost 2,000 people from around the world. I'll skip this a little bit for the interest of time. Basically, those were the teams that won, and this was really interesting.
I can say that, for example, two teams here are from the University of Michigan. One team, the top team, is from Fudan. One team is actually from my alma mater, where I postdoc at the Burnham Institute. And one guy, I think this one, is from a Japanese car maker that never has done any kind of bioinformatics in their life. And they jumped really high in the leaderboard in the last minute almost. So that was great. The solution write-ups are there for you to look at, and you can just Google Kappa5 Kaggle or just look at this QR code to find it. I'll skip the here. Let's see some of the results. So to have good machine learning results, you need kind of a good baseline. So the baselines that we use are BLAST, because BLAST everybody uses, so you're supposed to be better than BLAST to give some effort. Various embedding similarities. So the idea here, everybody uses large language models. So not large language models that are trained on human language documents, but large language models that are trained on protein sequences. That is what everybody was doing, at least everybody who reached the top level. So we looked at embedding similarities as the baselines. And these are precision recall curves. Precision recall curve is something that the better you do, the more you are up here like this. So these are precision recall curves for the results. The baselines are in these staggered, in these lines down here. So anybody doing better than a baseline is going up here. These are predictions in the biological process ontology, cellular component ontology, and molecular function ontology. So for example, for this protein here, we knew, for this group of proteins here, we knew nothing. This is the knowledge gained. How well did people predict it? For these proteins, we knew something in these two ontologies, but not here. And finally, for these proteins, new leaves were given. And again, these are the predictions. There's a lot to talk about here. I'm just showing you a sample of the results. You can ask me later about how we worked. The nice thing about Kappa5 is that if we compare the results and if we do a head-to-head competition between Kappa5 methods and the other methods, between Kappa4, 3, and 2, we see that Kappa5 top methods did a lot better. The orange point here in these distribution plots is BLAST, so you can see where BLAST is at. Kappa5 method did a lot better in the various categories. And the takeaways are basically we had a really great growth in participation and skill diversity. So the AI community that had nothing about bioinformatics really brought in the use of embeddings and protein large language models and optimized them towards improving the way we can predict protein functions. That was amazing. That was really great. We had a significant improvement in predictions in Kappa5, especially in BPO. Diversity of approaches may spur combined models using promising ideas from each methods, and partial knowledge challenging. I sort of alluded this, but basically the idea is to predict more leaves or more nodes in a Go DAG than there are right now. Really was a new thing, and it set an opportunity to harness known function for conditional predictions. These are the people who participated in it. A special shout out to Clara, who really did all the heavy lifting. We usually have one person who does the work, and that was her. I'd like to shout out for my two students, Parnal and Ann, who are here in the audience, and will be giving talks later. I thought that Silvio would be here, but maybe he's online. And Damiano and Silvio helped a lot. We just had to talk about DisProt, so we did that as well. And the people from Kaggle and UniProt, Sandra's here. She's also talking about, she'll be talking a little bit about this as well. And finally, to our funders, the NIH, and of course, the CAFA participants. Thank you. Barely a minute to spare, I'm sorry.
No, we have time for a question for Iddo. You want to come down?
Thank you. I'm interested about the improvement in the BP. So Paul showed us that the number of terms in BP, right, has been going down and you said that the biological process predictions were getting better. Do you have any sense if those two things are related at all?
Okay, going down is relative. BPO is still,
It's still
it's
big.
like saying that the population of India or China are going down. They're still the most populous nations on Earth. BPO is the most populous gene ontology
Yes.
aspect on Earth. No, I don't think it's related to that. I think what it's related to is the fact that Paul talked about homology transfer, but homology transfer works well in molecular function. It works much less well. It's not even a good baseline, in my opinion, for biological process. You know, you predict something as a kinase in a mouse, but then that kinase does something completely different in human, right? So I think that the improvement came from protein language models being trained on this problem of BPO predictions, and thus being able to solve it better. I think that's the improvement that we got, that we stopped relying on homology transfer and started looking more at embedding similarities, where these embeddings capture information that is related to biological process that sequence cannot. Parnal will be talking just a little bit about that also.
Yeah, no, I'd like
after
to
the
hear more
break.
about that.
Okay.
Yeah.
Okay. I think, is your mic on? I can hear you, but I don't know if the people online can hear you. You can say something and I'll repeat it.
Okay, I'm gonna go loud because it's flashing. So thank you, that was really an interesting talk. I wonder if we go back to your box and whiskers plots for a minute.
This is, okay, great, thanks. I think I heard you say that the CAFA5, what you rated here for the CAFA5 participants was the top performers? And that's what's shown, not the entire set?
Yeah, the top, I'm not sure exactly what Clara put here, but I think it's the top 50, maybe the top 100.
Okay, and for CAFA 2 through 4, it was all of the,
No, we did top versus top.
okay, that's what I wanted to know. Thank you.
Okay. Oh, just one more thing. Biocurators here, anyone who's working in annotation, if you want to talk to me about giving us test data for CAFA, we'd love to have what you can spare, meaning giving us annotated proteins that that you can, for at least for now, hide from the general public, but give to us so we can put that into test sets. Thank you very much.
One more, thank you. And Malcolm is going to take us through Xenbase.
Good morning. I'm Malcolm Fisher. I'm one of the biocurators at Xenbase. We're the model and database that services the Xenopus research community. And I'm going to talk about some new Synteny visualizations that we've recently put onto Xenbase and how we made them. So I'm going to talk briefly about why we care about Synteny. Here we have a picture of a recent paper that had several amphibian species, and they made some chromosomal level Synteny maps across them. The ones we're interested in are Xenopus tropicalis and Xenopus laevis. And you can see here, Xenopus laevis unusually has two genomes all to itself because of a historical species hybridization event. So what we have that we've recently added to Xenbase is some nucleotide level Synteny analyses that we did using LastZ, and we're visualizing using JPERS. And some higher level gene order conservation Synteny that we're trying to use to do some annotation work, mostly based on existing orthology calls and GFF gene order data. And we have a simplified viewer for that. So why do we care about Synteny? One of the main reasons is because our researchers that we work with care an awful lot about having their genes properly named and knowing what they're actually doing. We had some recent new genome releases about 5 years ago, and these were mostly from automated pipelines for annotation. So the gene models were there, but in a lot of cases, we lost the names. A lot of things were lokes or other placeholder names. So people could do their analyses, but they had no idea what their genes were because all they got was a whole bunch of lokes. So we've been working to improve this, and we've done pretty well for tropicalis. We've got like 6,000 or 7,000 genes that we've added proper stable identifiers for. We're having less luck with laevis because it's more complex because of that duplication, but we're still working on that. And we care about this sort of thing because it really helps us to get the names. If you know about the Synteny in a region, it helps inform your orthology calls. And if you know about pre-existing orthology, then it helps to know more about that region. And I mean, Paul said why orthology was so important. There are already a whole lot of existing Synteny browsers out there. You might be familiar with some of them. This is Genomicus, which has a huge amount of phylogenetic information and tells you about potential or imputed ancestral genomes. This is the MGI's multi-genome viewer that we like because it has both the L and the S, which obviously is very informative for us, and most other places treat those as the same. So again, UCSC has some Synteny information, and they also will only treat the Xenbase laevis as if it had like one long contiguous genome, and they don't let us treat the L and S distinctly. And similarly, the NCBI's comparative genome overview has relatively recently added the Xenbase species to it. It's got a very nice view, but it really is just giving you this linear comparison. So now you look at any sort of accessory data, and also doesn't split up the L and the S. So for our own nucleotide level Synteny, we've done a pretty simple approach. We've just taken the genomes we're interested in this case, Xenbase tropicalis and the L and S subgenomes of Xenbase laevis, and human at least to start with. We've just done some repeat masking for a hard mask. We've taken those masked genomes and run them through a series of pairwise alignment using LASZ, and then we take the resulting pairwise alignment files and we load them into JBrowse2 for visualization. And this is just one example. This at the top was from that earlier diagram. It shows you the LNS chromosome 3 from laevis, and you can see that there's a very large scale inversion there. And this is reflected here on our nucleotide level LASZ map. So what about the gene order conservation view? We're looking for like a sort of more quick and dirty, less granular overview that we can use to try and help us name genes in a region. Ideally, we want it to be sort of compact and have multiple species that we can look at the same time. We want to have a fairly simple and straightforward view of the nomenclature. We don't want a whole lot of accessory information. We'd like some identifiers that show us if we have existing orthology or paralogy statements. And we were lucky that we came across an already existing package called Simple Synteny that drew something like we wanted. So this is what you get from Simple Synteny. It's mostly designed for taking contigs and then giving it a set of genes that you're interested in. It will perform a blast and come up with all of the similar genes across those contigs, and it will show you a map of them. And it has some features. It tells you when you have inversions. It shows you when you've got large intergenic gaps. It gives you an idea of the size of the genome, or the gene, sorry, and their position relative to each other. We didn't necessarily want all of that, so we had a slightly different approach. Instead of using BLAST to derive our relationships, we used existing orthologs that we already had in our database, and we also fell back to simple symbol matching if we had to. And then we provided with a list which is either all of our genes or only a subset of the ones that we are interested in. We run it, it takes the GFFs and produces these smaller sort of 11 gene-wide contig maps. We then perform some optimization steps on those, and they're put into the Synteny drawer program that produces the images. And we have all the scripts we use to drive this on GitLab. I'm not going to go into this in great detail, but this is the format of the contig maps. And the main take home is that we have added this ortho group statement, which takes our pre-existing orthology assertions and adds them in as informing the relationships that are produced. We also want to try and align things better in cases like this large-scale inversion. Obviously, it's going to reverse the order of all of the genes potentially in a region. So we want to minimize this. So we've used a metric called the longest common subsequence, which is sensitive to inversion, but allows for gapping. So it's not as stringent as some other measures like Hamming distance. So we've calculated that for all the permutations of the different contigs that we have around a specific gene locus. And we then take the array of orders that give us the highest value overall. This is an example of that. Here you can see this Lavis L. There's a big inversion because this is flipped related to everything else. And after we do our optimization step, everything lines up much nicely, and you can see all of these orthologous genes here together. This is still a little busy for what we wanted. We wanted to make a more concise view. So we want to equalize the gene lengths, equalize all the contig lengths, remove all of the gap representation we were getting, give everything an equal intergene spacing. And also, where we have overlapping genes, we want to not show that overlap and just order them by their start coordinates. And this is just, again, so in the CMAP where we've normalized and standardized all of the distances and lengths of all of the elements we have here. And this is what we get as our final output. This is the actual web interface we have. Here you can put in your gene of interest, and it'll turn that as the center of the window, and you'll have the 5 genes up and down either side. And you can see here we have our nice orthology assertions. You can see where it's broken down because of the color scheme. And in cases like this, you have a tandem duplication here in Tropicalis and Lavis, and it's showing that these are all still part of the same ortho group. So at least beta versions of these are all now available on our site. If you go to Xenbase and go to the genome dropdown, then we have our simple Synteny search here where we just put in our gene of interest and return them. We also have links from our gene pages. And then we have our JBrowse2 instance, which has the Synteny, although I will say currently it has Minimap2 based Synteny because we're still finishing off doing our last Z analysis. This is the Xenbase team. Specifically, Knockley did all of the nucleotide level last Z analysis. Andrew Bell worked with us on developing the simple Synteny stuff. Vineet runs our JBrowse2 instance. Joe Wang did the web interface for the simple Synteny. And Christina did most of the testing because she was doing a lot of gene nomenclature review at the time. We're funded by the NIH Eunice Kennedy Shriver Institute for Child Health and Human Development. And I can take any questions.
Questions? Okay, well, let's thank Malcolm and all of our speakers this morning. Thank you so much.